Okay, well, welcome everyone. Glad you all joined us on this beautiful winter day. Our guest speaker tonight is Claire Schon, and her topic is a small solution to a big problem. As you guys already know that, or you wouldn't be here. We'll get to her in a moment. Just a few bits of club business to get through. Um, First on the agenda is Ontario Nature wants our support. They want us to sign on to a petition. And in particular, they want us to sign on in writing, in a printed petition, not an online petition. And it's a petition to the government of Canada. And in their words, it outlines concrete steps we can take as a nation to grow and thrive while protecting the air we breathe, the water we drink, the land we walk on, and all the species that call Canada home.
Bob, excuse me, but I believe this petition actually comes from the Y Marsh.
Yes, it does.
They're hoping Ontario Nature will jump on it too, but it's a Y
Okay,
Marsh project.
I was in error when I said that. So this is being generated by the Y Marsh, um, and basically there's a two-sided petition. I don't know if you can see this. Doesn't look like much. One it's the exact same thing. One goes to the Ontario government, one goes to the Canadian government, and basically it's calling on the government of Ontario or the government of Canada um, to protect and restore our natural heritage by keeping their promise to implement the 2030 nature strategy. And I've checked with Heather Carey at the Y Marsh. If you want to sign the petition, you can drop in at the Marsh. They have a copy at the desk. We're also, I believe you've sent this out to everyone. Have you, Ken? You can print
Everyone
this.
should have got a copy in their inbox, all of our
Okay.
members. And
Okay. So you can print this and circulate it around to all your friends. And trying to gather support, we're offering a prize for those of you who can collect the most signatures. And I'll show you here what the prize is. It's one of our lovely hats. I don't know. Maybe if I put it on my head, it'll show better. Anyway, this is just to encourage you. So do print off the form, circulate it, and then get it into the Y Marsh. And I've forgotten when the deadline is. The end of March 2026, I believe it is. So sign on and help us all. Um, a couple of other things. Some of you know that we're having difficulties with the website. My friend Wayne had been managing it, and there were issues, shall we say. We've resolved that. We were exploring other avenues to have a website. My friend Wayne has agreed to continue supporting the website, so that's a very good thing. But we are proposing some changes, and one of them is to move away from our .xyz website because, quite frankly, that's become associated with nefarious things like pornography and other scams. So what we're proposing is just not to change the website, not to change the club's name or anything like that, just to change the name of the website to naturemidland.ca and get rid of the XYZ extension. And if anybody has any strong feelings or thoughts about that, you can contact me by emailing me at mpfn.xyz. So info@mpfn.xyz. So there is that. Some of the club executive were invited by an organization called ROAR to a meeting. This is Resident Owners Against Radar, and they're talking about the proposed over-the-horizon radar array. Now, this is nothing that's truly new, but we didn't really understand what this was going to look like. And after ascertaining a few facts, quite alarming what they were proposing to do here. Now I'm going to show you some photos later on during the nature sightings thing that's going to highlight the extent of this thing. But basically, they're planning to eventually to cover up to 25 square kilometers with dipole antennas. So that's two posts like this, covering approximately 25 square meters of the Minising Flats. And the site has to be graded almost dead level. So that would mean deforesting whatever trees are left, grading it, knocking down buildings, and filling in creeks. Who knows the extent of it? So we're quite alarmed by that, and we've been invited to a meeting with, maybe Dorothy can help me out. I'm just finding out about this. Dorothy, could you unmute yourself and tell us about this upcoming meeting that you brought to our attention? Dorothy? I guess not.
I think I can. I think Dorothy set up something with one of the construction people that we may do an online meeting with. And again, this is all right beside the Minising wetlands in the Strongville Road area, one of our favorite places to go bird watching. Um, and I just remind you, Bob, that we have some of these Roar people that are going to probably come to our March meeting in person.
That's correct. And I have offered to draft a letter. I'm not certain to who, but we're quite alarmed by the prospect of this proposal. Not that we're against Canadian defense or against this radar proposal. We just think the particular location is quite inappropriate, especially considering the proximity of the military base at Camp Borden. So I think it's attractive to them because it's a fairly flat site that will minimize the grading that they would have to do. But you all know that this is one of the favorite places for birders to go for winter birding. It's a wintering grounds for snowy owls, and many of the grassland birds reside there in the summer. And as Ken pointed out, it's immediately adjacent to the Minising wetlands, which is of vital importance. Thousands of trumpeters or of sandhill cranes nest there, trumpeter swans, all manner of wildlife rely on that wetland, and filling in the creeks and the drainage could have some alarming consequences. Anyway, we'll keep you posted on what we learn about that. I wonder, is Anthony with us? Is Anthony?
I don't.
Okay, well then this is just
him on the meeting, Bob.
Okay, this is just a reminder that the club has an outing at the Martin Valley Tract at 10 a.m. on Saturday morning. The weather looks good. And it's a snowshoe outing, so bring your snowshoes.
It's Sunday morning, Bob. Sunday morning.
Is it Sunday? I beg your pardon. I thought it was Saturday. Thank you, Ken.
Sunday, 10 a.m.
Okay, so anyway, there is that outing. And a reminder that after the Q&A, when Claire is all finished, we'll do nature sightings, and I'll show you a bit about these radar arrays. So that's all I have. I'm going to turn it back to Ken to introduce our guest speaker. Ken? Not having much luck tonight.
You need to unmute, Ken.
Ken?
Ken.
Okay, is that better?
Yes.
Too many buttons to push. Just a few things about how we run these meetings before I introduce Claire and turn it over to her. If you have questions during Claire's presentation, put them into the chat, and then we can read those out at the end of Claire's presentation. When we get to the question period, if you want to unmute yourself and ask your question in person, you can do that too. You just click your little microphone button and ask your question in person. The other thing I always mention is that when we have our in-person meetings, we usually ask guests, non-members, to make a small contribution, $5 or something like that. a little more difficult to do when we're on Zoom, but not impossible. Anybody who feels inspired can make an e-transfer to the address you see above my head, treasurer@mpfn.xyz, and that will go into our accounts with no need for a password or anything like that, and we'll promise to do something good for nature with it. Okay, let's tell you a little bit about Claire Schon. Claire is a PhD candidate in the Waterloo Wetland Laboratory, led by Dr. Rebecca Rooney at the University of Waterloo in the Department of Biology. You can tell me if anything's out of date here, Claire. And her research focuses on botany, wetland ecology, and invasive species control, working to control invasive Phragmites within southern Ontario. And I understand Claire is not in Waterloo today. She's joining us from Haliburton. Maybe she'll tell us what she's doing there. But with that, I'm going to ask Claire to take over. And I'll unpin myself.
Okay, thank you. Can everyone hear me? I'm going to share my slides, but I just want to make sure everyone can hear me. I see some head nods.
I can hear you fine. Everybody else okay?
Perfect.
Yep, thumbs up from Vicky.
Okay, thank you. I'm just loading my slides. Computer's a little overwhelmed, 41 people. So I'm just going to try to load these now. But yes, I am joining from Haliburton. It's reading week at the University of Waterloo.
Claire, we've still got your presentation screen up. We're not full screen yet.
Yeah, I pressed the button. It's just still loading. I think my computer's a little overwhelmed right now, so I'm just getting it. Oh no. Well, this went seamlessly when we practiced it earlier, but of course, now that we're all here, it's not going to work. But I'll just give it a second before I try again. Oh, okay. Loaded on my end. I think you should all just see the slides now. Perfect. Okay. Thank you, everyone. Okay, I'm going to get started. Ken kind of covered it. My name's Claire Schon. I'm a PhD candidate at the Waterloo Wetland Laboratory. And I'm going to be talking about a very small solution to a very big problem, Phragmites. Maybe some people have heard of it, but I'll jump in. Before I start, I'd like to give a little introduction to myself. So I think we covered the basics, but just how I got into this field. My path to becoming a wetland ecologist, it was actually very linear, although at the time it certainly didn't feel that way. This is a photo of me in high school where one of my high school projects was to create a management plan for an invasive species. And my group we chose purple loosestrife, this invasive wetland plant. And the way we decided to control it in this high school assignment was through biological control. So biological control is the use of a living organism to suppress or control another living organism. In this case, there's beetles that feed on purple loosestrife leaves. And at the time when I was in high school, I thought this was a really fascinating idea. I thought it was a really cool use of our understanding of ecology. And ultimately, it was this project that really sparked my interest and made me want to pursue wetland plant ecology. And then the other thing I want to say before I get started is that, I work on a really big team with a lot of really amazing folks. And I've tried to include all their logos here, but I work with a lot of conservation authorities, a lot of conservation organizations, as well as other researchers. And this is part of a really large Phragmites project. But I'm just really grateful to work with all these people and have a lot of support. So a big thank you to all the groups that you see here. So a bit of a roadmap for what I'm going to be talking about today. I'll cover quickly what is Phragmites. Sounds like maybe some people in the group already know it, but I'll just cover that briefly. What are the impacts of Phragmites? How do we control it? And what is biological control? And what is Phragmites biological control? So this invasive plant that I study is called Invasive Phragmites. Its scientific or Latin name is called Phragmites australis, subspecies australis. Its common name is European common reed, which gives you a hint to where it's from. It was introduced to North America in the 19th century from Europe. It's this perennial wetland grass, so it lives year to year in this below ground network of something called a rhizome. and it can grow really densely both above and below ground, sometimes over 100 stems in a single meter squared, so 1 meter by 1 meter area. But my personal record for how many stems, I think, is 153 stems in a meter squared. So it grows very densely. My supervisor, Dr. Rebecca Rooney, likes to say that walking through it is like walking through wicker furniture, which if you've ever tried to walk through Phragmites, I think that resonated a lot with me. And it's also very tall. It can grow over 5 meters tall, which is about the height of an adult male giraffe. And so I know you can't see me in person, but I'm pretty short. And so I do like to say that I'm one very short ecologist battling one very tall plant. To give you a bit of history on Phragmites invasion in Canada, these are looking at herbaria records that we had samples from, and they were able to determine whether they are invasive Phragmites. And so our first Canadian record of invasive Phragmites is from 1910, right on the east coast in Nova Scotia. Very quickly it made its way to Quebec by 1916, and then it spread throughout the St. Lawrence River into the Great Lakes region, and by 1948 we have our first record in Ontario. Very quickly in the early 2000s, by 2004 it's made its way to Northern Ontario, and by 2000 and 2003 we have our first sightings in British Columbia. And it was actually in 2005 that Paul Catling from the Agriculture and Agrifood Canada named this Canada's worst invasive plant species. And it really has continued since then. By 2009 it's made its way to Manitoba, actually where I'm originally from. And by now it's spread to all 10 Canadian provinces and represents an emerging threat to the prairies. As you can see from this map here from iNaturalist data, observation data posted online, we can really not even see Ontario. It's well within its established range. And Ontario is kind of, at least Southern Ontario is just buried by all of those observation dots. So I've been talking about invasive Phragmites, but I do want to specify that in North America, we have actually two what we call lineages of Phragmites. And the first one has been on this continent for a very long time. This is known as Phragmites australis subspecies americanus. And this is our native Phragmites, our native subspecies of Phragmites. And one of the reasons, actually, I find this very interesting, the reason why we know it's native, we have a lot of evidence, but one of the most interesting reasons is actually from the Shasta ground sloth, a long extinct large sloth that used to roam North America. And fossilized feces of this sloth have been found and dated to about 40,000 years ago, and it was found that it actually ate this plant species. The more recent addition to the continent is the one on the right and the one that I study, Phragmites australis subspecies australis, or the European introduced version of Phragmites. And so I'm going to be talking about this invasive Phragmites for the majority of the talk. I actually looked up this map, this region looks familiar to many folks, but I did look up because when I give this talk usually most people have no concept that there is a native Phragmites, or they've never seen it or never heard of it. But I did look, and it looks like in your area you have both invasive Phragmites in those pink dots and native Phragmites in those blue squares. So there is a bit of both in the Midland area, and so that's really encouraging to see that there is still some native Phragmites kind of kicking around. Good to see that you have both. People do ask me all the time, how does it spread? And the short answer is that humans have been very instrumental in helping Phragmites spread. It can propagate itself via seeds. It's a plant, of course, so those seeds can get transported, they can germinate elsewhere, and then start a whole new life somewhere else. And it can also propagate itself by small cuttings of either its rhizome or stolon. So a rhizome is a below-ground storage organ that stores resources for the plants. And so a cutting of a rhizome can get cut off and again start another plant somewhere else. And then what I have in this photo here is actually a 15-meter long Phragmites stolon. And you can see that about every 10 to 20 centimeters along its length, it's actually sending up and starting a new stem. And this is another piece that can get cut off or maybe break off and float downstream, and again start another invasive Phragmites patch. So if any of these things were to uproot or get caught in machinery, be transported knowingly or unknowingly, it can all create a new plant and really helps Phragmites spread, and has really helped it spread so rapidly. So when Phragmites invades, it causes a number of ecological impacts to the wetland habitats that it invades. Wetlands are able to support so much biodiversity because of their zonation, meaning that plants form these distinct communities at different water depths. In the deepest part, also the right side of the screen, we see that there's kind of submersed aquatic vegetation floating throughout the water column. And as it gets shallower and shallower and shallower, we see different plant communities at these different water depths. And so we see like floating, we might see pond lilies, we see cattails or bulrushes, which maybe some people recognize. And then we get even shallower, and a new zone of meadow marsh plants are out of the water, but still getting their roots wet. But this really changes when Phragmites invades. We tend to get just one Phragmites zone, and we lose this zonation. It tends to just be this one monoculture Phragmites, and it really seems like there are no limits to where Phragmites can grow. So when we get this frag invasion, we not only lose biodiversity of wetlands, not just including plants like seen here, but also birds, reptiles, amphibians, macroinvertebrates. In addition, we also lose a lot of the wetland services or wetland ecosystem services that wetlands provide. So it alters how wetlands functions, it accelerates both the carbon uptake and then the release of carbon, so it's not beneficial in terms of carbon storage and climate change. Phragmites is also very effective at outcompeting native plants for really important resources for plant growth, like nitrogen. It's able to grab it faster and more efficiently than other native plants would be, and it's also able to intercept a lot of light. So in this photo I have on the screen, it's actually like a clear, blue, sunny day, and I'm looking straight up, but you really can't tell because I'm under the Phragmites canopy. It forms this dense canopy and it shades everything below, so native plants really struggle to stay alive in this understory. It also mutes the natural fluctuation in temperature that some species rely on. And it has become a huge threat to biodiversity in Ontario. An analysis of over 200 imperiled or species at risk in Ontario done for the Ministry of Natural Resources concluded that 1 in 4 of our species at risk are directly threatened by Phragmites invasion. So this accounts for like 71% of our turtle species at risk, almost 30% of our native plant species at risk, and about 20% of our bird species at risk directly list Phragmites invasion as one of their causes for being threatened. It also has a number of economic and societal impacts. So Phragmites grows in a lot of ditches, maybe you've seen it on the roadside, and it impedes the drainage of these agricultural ditches. It reduces property values. You can imagine if you have a very beautiful waterfront property and all of a sudden Phragmites invades, sometimes you can't even see the water, and you won't even know it's a waterfront property anymore. It impedes opportunities for recreation. You can't really fish in it. It's not good fish habitat. You can't kayak or do any of those sorts of things that you might in another wetland. And of course it affects things like birding, which I know we were talking about before this meeting started. And it also becomes a huge fire hazard as well as a traffic hazard, which this photo shows really well of it blocking a traffic sign or also just blocking roadway visibility in general. So hopefully at this point of the talk, I know I went through it really quickly, but I hopefully you understand that there's a need to control Phragmites. We simply can't risk letting it continue to spread and continue to take over our wetlands. And I'm kind of lucky Phragmites control is pretty well agreed upon. That's a worthwhile endeavor. We have lots of support from not only those partners listed, the provincial government believes really strongly in Phragmites control. They've invested a lot of money very recently for Phragmites control. But one thing I really want to highlight before I move on for controlling Phragmites is that it's incredibly important that we ensure that we have the invasive Phragmites and we're not taking any action to harm or control our native Phragmites. So understanding that there is two subspecies and knowing how to distinguish them is a really important step before you decide to actually control Phragmites, because we wouldn't want to harm our native plant species. But in terms of controlling Phragmites, there's a lot of effort in Ontario specifically. Some groups have this catch line, which I like to say and share, that they want to be frag-free by 2033. And when we think about invasive plant management in general, we kind of have 4 options typically. And just in general, we have chemical control, the use of herbicides, mechanical control, or the physical removal of the invasive plant. We have biological control, which I mentioned earlier, and of course I'm going to come back to. And then we have our fourth kind of option, which is do nothing. For a lot of non-native plants, this is the reality. There are a lot of invasive species, there are a lot of non-native species, and we don't always have the effort. But hopefully I've convinced you that by this point that like doing nothing for Phragmites poses a real significant risk to the environment. In terms of Phragmites control specifically, chemical control is the most widely used option right now. That's using herbicides, and they can be applied via a number of ways. Applied via helicopter when you have really large swaths of Phragmites, like they did in Long Point. If people are familiar with that area, it was hit really significantly with Phragmites invasion. And so one tool that was used extensively was helicopter aerial application. Amphibious vehicles can also be used on the ground via backpack spraying. And then one method that's kind of a little bit still futuristic right now is drones. That's been some active area of research in the Waterloo Wetland Lab that I'm a part of. But in general, chemical control can be really effective. You know, if done correctly at the right time, it can remove over 90% of Phragmites biomass, sometimes even up to 99%. So very effective. And this photo I have of here, that speck in the middle photo is actually me in the middle of a huge swath of Phragmites that had been treated with herbicide. And I was actually joining my colleague from some of the work that they lead to conduct plant surveys to see what plant species grew back after herbicide treatment. So this isn't work that I lead, this is work that my colleague has generously shared with me, and it's allowed me to present and share with you all. But we have been tracking in the Waterloo Wetland Lab, work led by my supervisor and also led by a PhD student, Jersey, what happens to the vegetation communities after Phragmites is treated with herbicide. So in 2016, before the treatment took place, they monitored the plots and found, of course, they're all dominated by Phragmites. And then in 2017, you see these orange bars, and those orange bars tell us that those plots, those areas where formerly Phragmites dominated, were now dominated by different non-native plant species. So not Phragmites, but a different non-native plant species. So initially, that's kind of discouraging. You don't really want to replace one invasive species with another. But over time, you see the rise of those green bars to native-dominated communities. So that means that native plants do return. It takes multiple years, at least 4 or 5 years. And this is in the best case scenario kind of work in Long Point, Ontario, where there's a really good seed bank, but really good outcomes here for this Phragmites control program. One thing I'll just say is that, it's very effective, but chemical control is also really costly. It was estimated in a 2019 study that for Ontario alone, it would cost upwards of $109 million. And considering that Phragmites has continued to spread since 2019, and just the cost of things is raising, that might be a little bit of an outdated number by now. The other thing is that you really need to continue treatment and have the funding to continually go back to these patches of Phragmites and kind of spot spray or kind of clean them up year after year to make sure you're really trying your best to eradicate Phragmites. Some mechanical control methods. One of the most widely used method is called cut to drown, and it's used extensively in the Great Lakes region, where stems of Phragmites are cut below the water to really drown out that rhizome or that below ground tissue and drown out the Phragmites plant. This can be done using a machine here, like you see. This is actually the Invasive Phragmites Control Center, who's been leading the charge for a lot of Phragmites control across the province. And it can also be done via raspberry cane cutters or basically essentially by hand. Another method, it involves cutting the Phragmites stem below the ground and severing the rhizome, again that storage organ, from the stem. I like to promote this method, especially to community groups, because it's really accessible. All you need is some really willing volunteers and a couple shovels. And it has just limited tools, and of course can be done with drier spots where you couldn't do that cut to drown method. And this has been pioneered by Professor Lynn Short at Humber College. So another great method for controlling Phragmites, again, manual removal. But these kind of highlight some of the gaps that we have in Phragmites control, that we need a really long-term, sustainable, and cost-effective tools. Chemical control is really effective, but it's also really costly, and sometimes it might pose additional risk to the environment, especially if you're not doing it so carefully. And then mechanical control is really labor-intensive. It takes a lot of really dedicated people, but there's a lot of frag across the province. So this is where we kind of arrive at the idea of biological control, or biocontrol, or it's abbreviated to biocontrol. And so this is an alternative to chemical or mechanical control, and biocontrol uses living organisms to suppress a different living organism, and in the case of weed biocontrol, we are trying to suppress an invasive or weedy plant, which we call the target plant. And the living organisms that we use to suppress the target plant are called the biocontrol agents, and these are the host-specific natural enemies of the target plant, and so these are the natural predators of these target plants. And so the essence of biocontrol, the core of it, is reuniting two species which are natural enemies of each other. And the goal is to achieve a gradual long-term suppression, although it's not an eradication of the weed populations. And a really successful example, maybe some people are familiar, if you know any biocontrol program, hopefully it's the purple loosestrife biocontrol program, where in the 1990s, purple loosestrife was an invasive species, and it still is today, but there are 4 biocontrol agents approved in Canada to control this invasive plant. There are 2 species of leaf-feeding beetle, as well as a root weevil, and either a flower or seed weevil. And so these species work together to help control purple loosestrife, and they're still active across the province. Biocontrol has a number of advantages. It's that we select host-specific insects or agents that only feed on the target plant. So in the case of herbicide control, herbicides typically aren't selective. They do not discriminate, and they'll essentially kill any plant in its path. But with biocontrol, you're intentionally selecting insects that only feed on your one target species and that species alone. It's very cost effective, and its cost efficiency stems from the fact that agents can form these self-sustaining populations where they complete their natural life cycle and continually suppress the target plant population. And another related advantage is that they can fly or disperse to new patches and suppress more and more populations of this target plant. And so with that, I'm going to talk about the development of the Phragmites biocontrol program, which began in 1998. And so one of the first steps in any biocontrol program is to figure out what natural enemies of your target plant are. And luckily, in Europe, Phragmites has a lot of natural enemies. There are over 60 herbivore species that feed exclusively on Phragmites. So they don't feed on any other European species. And so they feed on nothing else but Phragmites. And so there was a huge list, but eventually a promising group of species were identified, and those are stem-boring moths. And so what we know about these 2 species of moths. They don't really have fancy names, unfortunately. They only have their scientific names, which are Arcanara neurica and Lanisa gemnopuncta. And we know from Europe that they only feed on Phragmites. They cause significant damage to the plant, and they were extensively tested over a 11-year period to determine, after extensive testing, like I mentioned, and a peer review process, that they're host-specific, what we call host-specific to Phragmites australis, meaning they need Phragmites to complete their entire life cycle, and they demonstrate a really strong preference for the invasive Phragmites, and so the risk to native Phragmites with this method is also very low. And the reason why they're called stem-boring moths is because they bore in and out of the stem. So you can see those 2 little holes there on the Phragmite stem. Those are what they call their bore holes. That's how they get inside and outside of the Phragmite stem. And to kind of review the life cycle of the entire, the stem-boring moth is completed within patches of Phragmites. And so the first kind of life cycle or life phase that I'll talk about is the caterpillar life phase on the right there. The caterpillars feed on about 2 to 4 Phragmite stems during this life phase. Their next life phase is called the pupa life phase, and pupation occurs within the lower half of a Phragmite stem. And they're in there for about a month, and then in about late summer, early fall, these adults emerge, and the adults are very short-lived, and their only focus is to find a mate and lay their eggs. And then the eggs are laid on the leaf sheaths of Phragmites on a part of the plant, and they're kind of tucked within the plant. And then it's actually these eggs that overwinter, and then again in the spring their life cycle continues, and it's these caterpillars which are I like to call them the very hungry caterpillars. I'm sure many folks are familiar with this story, this children's book. And unfortunately, these moths aren't quite as colorful, but the very hungry caterpillar, this like bright green caterpillar, but unfortunately these are more of a brownish cream color, but they're the ones doing all the hard work. And of course, they're very hungry for Phragmites stems. As the larva or the caterpillar is born to the stem, they're feeding on the inner stem tissue. They're damaging like the vasculature, like kind of like the veins and artery of the Phragmites plant. And when we see stems that have been attacked by the caterpillars in the field, we kind of see one of three responses. So on the left, I've drawn like an undamaged or like healthy stem of Phragmites. Then the second one over, after the caterpillar feeds on the stem, especially early in the season, the stem will die. With this wilt-only option, just the top half of the stem wilts and dies, while the lower half of the Phragmites stem remains alive. And then in this branching option, one of the outcomes of this feeding is where the top half of the stem originally dies, but then it kind of tries to reroot. It's like vasculature, it's veins and arteries, and so it produces more leaves on these side shoots or secondary branches. And what we know from Europe is that this reduces the height, this reduces the above ground biomass or the productivity of the Phragmites plant, and also the flowering, so the ability of the plant to produce seed. And the goal of this program is to reduce the competitive ability of invasive Phragmites and kind of thin the Phragmites canopy, allow for more light to reach the ground, and allow for plant community recovery to occur. So I'm going to be talking about this Phragmites Biocontrol program, and in 2019 a petition was submitted to the Canadian and American federal governments, and the Canadian federal government approved the use of these two species of stem-boring moths as biocontrol agents in 2019 as well. And the first and only pilot program was started by my research collaborators, mainly based out of the University of Toronto, as well as Agriculture and Agrifood Canada. And they've shared this map with me, but now, about 7 years after federal approval, they've managed to release biocontrol agents at actually 60 sites across Ontario, mostly within southern Ontario, but we also have some sites at North Bay. And so this is kind of the release side of things. I'm on the research side of things, so I'm not too involved, but I do work at a number of these sites, or a small select number of these sites, to learn more about these moths and their effect on the Phragmites plant in North America. And so I think for this presentation, I'm going to try to answer these couple questions here. The first one is, are these biocontrol agents surviving? Are they able to overwinter and live in Ontario? Are they dispersing? Are they moving throughout the landscape? And what are the impacts to Phragmites? That's the question I'm most interested in. And then I have maybe a small short story to share at the end. And to answer the first question, I'm going to talk in detail about some releases that were conducted actually on the University of Waterloo campus, again, where I'm located. And so in 2022, my collaborators released biocontrol moths into a large patch of invasive Phragmites, and we had two kind of release methods that we were trialing at the time. And so one of them in that middle photo is called an egg cup, and if you see in the top right photo, those little squiggly lines, those are all actually the very, very, very newly hatched caterpillars of the biocontrol agents. And so those hatch in those egg cups. It's got a mesh bottom, and so they'll kind of crawl down that stem and then go on and eat their first Phragmites stem and continue to eat those Phragmites stems. The other method is a little bit more intensive, where the eggs are actually hatched in the lab, and then they're individually transferred into a cutting of a Phragmites stem. And so that gives them food for their first part of their life phase. And then afterwards, they can leave that cutting of the stem that's in the bottom right corner. They can leave that and then start to eat their first Phragmites stem out in the wild, essentially. And so we were interested in these two release methods. And these were methods that were pioneered by my research collaborators. And it was really just a way for me to kickstart my research field site. And that's actually when I joined the project in 2022. And so in terms of answering the question, are they surviving, I'm going to jump to 2023, so 1 year after the release. And in early 2023, one of the first things I saw was this Phragmites stem that had just came out of the ground. It was early May still. And then I kind of picked up that stem and I was curious what was going on with it because it looked kind of sickly. And I opened up the stem, I kind of peeled back some of the layers, and I found my first caterpillar out in the wild. So I know that they made it at least 1 year after we released it. And then I wanted to continued my field season that summer. And then I saw what is evidence of a pupa currently in the middle of pupation. So I don't know if you can see that kind of transparent circle on the Phragmites stem. It actually kind of chews through the stem and leaves itself what we call a little window pane. And then that adult, once it's done pupating, can actually push through that window pane, that really thin layer of plant tissue, and then the adult can emerge. And I will say that this is a photo of one of the Biocontrol moths, and this is, I think I'm the only person to ever see them in the wild. They're very short-lived. They're actually usually nocturnal moths, but I happened to be at the right place at the right time. I don't think this might have been a once-in-a-lifetime encounter, but I did have an adult moth land on me, and then it continued on. I was able to snap this photo, and this is an incredibly zoomed-in photo of the adult moth. And then, well, I just really wanted to finish it off and get the last part of the life phase, so I started looking in late summer for eggs, and I found them. Again, this photo is incredibly zoomed in. They're only about 1 millimeter wide. They're very tiny, and they are laid underneath the leaf sheets of the Phragmites. And so I also found the eggs that late summer. And so I like to call this a little bit of biocontrol bingo. I finally saw all 4 life phases, and so I know that at least one life cycle was completed, but I will say that they've continued on at this field site and at all my other field sites. So they are continuing to survive. So that answers that first question. And then I was also really interested, are these biocontrol agents, are they dispersing? Are they moving throughout the landscape? Are they finding new patches of Phragmites to attack and feed on? And so we were kind of looking the next field season again, 2023. I was interested in knowing where did they go from my initial release point. And so I found some patches of Phragmites, and I ended up surveying about 6 patches where you see those dots on the map. And those were within a 500-meter radius of my initial field site. And I actually detected that the moth had already traveled to 3 of the 6 sites that I checked, a maximum of 480 meters away, which at the time, it was really encouraging, and I like to call it one giant leap for moth kind. And then the next summer, I had just a little bit more time to survey the patches of Phragmites and to see if there was evidence of the moths traveling and dispersing to those patches. And I just couldn't stop finding them. We searched over 30 sites, and we found them at most of them. I think it ended up being about 25 sites. And this year, in 2024, we found them a maximum of 2.1 kilometers away in about 2 different directions. And so that was really awesome. Unfortunately, I don't know if they traveled any farther than that. We're kind of off the University of Waterloo campus. I didn't really have permission to sample at those locations anymore. And I was really encouraged to see that, at a minimum, they're traveling 2 kilometers into patches of more invasive Phragmites and eating Phragmites there. In 2025, just this past summer, I returned and I tried to do the same kind of sampling. And I found that they're staying at those sites. They're persisting at most of these sites. So not only the moths getting there, but they're also staying there. And so that was really encouraging to see. And then I was also able to directly compare the data that I had from 2024 to 2025. And I found that in general, they're increasing at those sites. So they're attacking more Phragmites stems in 2025 than they were compared to the year prior. So we have some green on the periphery there. A couple sites where it decreased a little, but in general, they're increasing at most of these sites. So that answers my second question. We know they're dispersing on the landscape. Not only they're surviving, they're dispersing. And my next question, which is really about the heart of my PhD, is what are the impacts to the invasive Phragmites? And so I start at the kind of smallest unit, which is looking at a single stem. And those are single stems that have been fed on by the biocontrol agents. We know because we can see those boreholes on the Phragmites stem. And so what are the impacts when the caterpillar feeds on just a single Phragmites stem? And then how does that translate to entire plots or the stand of Phragmites? And so the first thing I'll say is this is a different research site that I'm going to be focusing on for a little bit. This is called Rare Charitable Research Reserve, kind of in the Cambridge area of Southern Ontario. And again, we released moths there in 2022, and we've been tracking them ever since. And we've seen over time that they're attacking more and more stems. Those moth populations are growing, and we're seeing year over year that they're eating the invasive Phragmites and working to control it. So you can see it starts off, this is 2023 was the first year where the moths were on their own. We didn't interfere. We were just monitoring what happened. And initially it was very low at the one site. We had a wet site and a dry site, and we were kind of comparing them. It was initially 5%, and 24%, which means 1 in 4 stems were attacked by those biocontrol agent caterpillars. Then that increased dramatically in a really good way in 2024, more than half at the dry site and over 80% at my wet site. So really, if you looked at 5 stems, 4 of them would have been fed on by those caterpillars. And it continued to increase in 2025 as well. Although the one site took a little dip, but that's because there's just so many stems, it's hard to really feed on them all. And then I was also interested, well, if I harvest the stems, if I bring them back to the lab and start measuring some things, I want to quantify, I want to know what's happening to the individual stem. So that's what I did. I brought stems back to the lab, I harvested them, and I measured a whole bunch of things. I'm going to try to walk you through these graphs, but the biggest things that I noted was that the stems are shorter. This is their height in this panel B in the middle here. So the stems are shorter. They're producing less leaf area, which leaf area is really important because that's how the plant photosynthesizes. That's how it, makes its own resources. So it's producing less leaf area to photosynthesize from. And it's producing, if we go to the bottom right there, it's producing less biomass in all. And our dead stems, the stems that died after the caterpillars fed on them, are lower than our that middle box there, the branching stems, the stems that produce those side shoots. But both of those are lower than our control or our healthy stems for comparison. So really good signs there, and that tells us that those stems are showing an immediate response to the biocontrol agent damage. So those caterpillars feeding reduces the stem height, it reduces the biomass of the stem, the leaves, and the whole stems. And all in all, that branching, even though it's trying to kind of compensate for the damage, it's not able to fully compensate for the damage to the main stem. And then I wanted to know, well, what happens to entire plots? And so you can see all those kind of pipe cleaners there, all those colorful pieces. Those are me marking all the stems that have been fed on by the caterpillars. And I wanted to know what happens when there's a lot of caterpillar attack stems in the plot. What happens to the actual plot? And in this first figure, A here on the top left, you'll see that the canopy height, or the height of the Phragmites, decreases. In the middle, panel B, the biomass decreases, so all in all, it's affecting the whole productivity of the plant. The number of live stems is reduced, of course, because a lot of the stems will die when those caterpillars feed on them, so there's less live Phragmites stem. And then we start to see a little bit, although it's not super robust relationship, but we do start to see that maybe a little bit of light is starting to reach the ground. But it's still pretty strong. It's still blocking a lot of light reaching native plants. So that's really another positive sign. But if you want to see some more photo evidence, this is my site just 2 years apart, or 3 years really, taken at the end of August in 2022 when we had just started releasing biocontrol agents versus 2024. We found that you could really see how much they had suppressed the entire stand of Phragmites. If you see that that brown layer, that's all the standing dead stems from previous years, and those green stems are getting much shorter. It's reducing the whole canopy. And I saw that again this year at my site again. So 2024, they kind of didn't eat from this one patch or this one area of the patch, but in 2025 there was a lot more of them there, and there was again that whole layer of dead stems where it didn't grow any taller than that. So we know that that individual stem damage does start to scale to the stands, and particularly the canopy height shortens. And so the canopy height shortened a lot. It gives a really a pathway for native plants to recover. Okay, I'm going to jump into a bit of a story here, but I will talk about some of the monitoring that I've been doing as well. So I'm really interested in what's happening to native plants in response to Phragmites control. And so I have been monitoring over the years, although I should say biocontrol works over a really long time span, sometimes up to 2 decades. So my 5-year PhD will not even cover the beginning of that. But I am setting up a monitoring program to hopefully capture some of these changes so we can answer questions like, does native plant abundance increase? Does native plant diversity increase? Do we start to see new invasive species enter? Hopefully not. And are there certain types of plant species that benefit? So when I'm out in the field, it looks a lot like this. I'm in the patch of Phragmites, but I'm counting all the native plants. I'm identifying and understanding what native plants are there. But also monitoring how abundant they are, if that's changing over time. And I work in some really beautiful sites. Here's just a collection of plants at some of these sites. And sometimes, I'm sure this secret will stay in the room, but I tell my supervisor that I'm just going to check on the moths. But I just happen to find the perfect day when the irises are in bloom. And I just get to appreciate all the native plant diversity that are at these sites. So no one tell my supervisor, but I sometimes say I'm just going to check on the Phragmites, and I walk right past it to look at what's surrounding actually the Phragmites. And so one thing we wanted to do, instead of just passively monitoring, which we're also doing, and the kind of watch what happens after we release the moths, I also wanted to conduct a more active experiment to ask the question, do native plant species benefit from Phragmites biocontrol? And our goal was to understand if there are really mechanisms in place for native plant communities to recover following the release of those biocontrol agents. And I guess the details of this aren't so important, but we selected 4 different native plant species that had been previously studied as competitors of invasive Phragmites. And so we knew from a range of competitive abilities, we selected these 4 plant species. And then I went out and I planted 416 plugs of these native plant species. I got them from a native plant nursery. Maybe you recognize some of these. This is Allegheny monkey flower. We have fallow manna grass, bristly sedge, and also spotted Joe pieweed. And we planted these at 3 wetlands in Ontario, and we were interested to see if there was a measurable effect or benefit when there was biocontrol. And the site that I had done this most intensively at and set this experiment up at was my University of Waterloo field site, where I basically, there wasn't much oversight. I kind of had the freedom to load as many native plants into this wetland as I wanted. So I got a big sample size from this site. It was easily accessible. And we had also released a large number of biocontrol agents like we saw in those release method photos. And we were right, there was still a lot of biocontrol agents there. It's about a 4,000 meter squared site. And in the first year, we were really actually hard-pressed to find any areas where the moths hadn't already reached. And so I was really excited this. I was feeling great. And I was also 3 weeks away from my PhD comprehensive exam, this really important exam that PhD students take. And I was so excited for my fieldwork to begin. And I had this moth population that I was building over 2 years. I was just doing a lot of fieldwork. I was really getting to see all these beautiful wetlands. And I had 49 plots dedicated to monitoring the plant community changes over time. And then all of a sudden I get this really weird text that says, "Hi, I'm so sorry to hear that your field site burned down." And I thought that text was pretty odd because this is in the middle of urban Waterloo. This, not a site that should be ravaged by wildfires by any means. But it was a really strange text to receive, and so I went to my field site pretty much right away. And sure enough, my lovely, lovely field site where I had all of my experiments had burned to the ground from unknown causes at the end of April in 2024. And while that was pretty devastating, including the loss of my native plant experiment, so that photo on the right and what I have circled is the small electrical flags that I used. And the only thing left, I used those to mark my plots. The only thing left of those was that kind of crispy piece of metal that was really tiny. I had to get on my hands and knees just to find them. And I found these, burnt pieces of electrical flag metal. One thing I did find though that kind of was hilarious was I had lost a meter stick in the Phragmites, probably more than one, but I at least found one, and I found it and it had burnt almost all the way to the ground. I think the last 10 centimeters of it weren't quite burned, but that tells you how intense the fire was at this field site. And so, well, I was like super devastated that my experiments had completely unraveled. I just had a realization that when life gives you lemons, you should try your best to make lemonade. And so that came in the form of generating new experimental hypotheses, experimental questions. And so yeah, that experiment was kind of done, but we were able to ask some new new questions, like how does Phragmites respond to fire, and did the fire harm our biocontrol agent moth populations? And so this photo maybe is a little misleading because the field site was about 4,000 meters squared, but only 3,000 meters squared of it burned down. There was a small area that was actually right across the street was a gas station, so the firefighters who got called to the scene, it seems like they protected that area right close to the gas station. And then there was kind of another corner opposite this corner that you see here, where again the Phragmites didn't burn. And even though it looks like a lot of the site burned down, I was very lucky in that actually only just a little bit more than 50% of my plots that I had monitored for 2 years prior, only about 50% of those burned down, and then the other half remained intact with this remaining Phragmites standing dead material. And so we had actually a comparison. We knew exactly where the fire margins were, and we can compare the burned and intact plots. And it wasn't a surprise, Phragmites grew back very, very quickly. This photo is just 3 weeks later at the same field site, where you can see the Phragmites is just starting to emerge, and that's what that burned area looked like just 3 weeks later. So of course this was May 2024. I have a photo from August 2024, and I don't know if you can spot me, but my head is in the middle there. That little speck is actually me standing in that same burned area just 4 months later. And so we were very lucky that not only did we have 2 years of monitoring, we knew what the Phragmites was like 2 years prior to the fire, but we actually kept doing that work, kept monitoring. And so the first question is, how did the Phragmites respond to the fire? And so I have this figure here where on the left side is our before fire 2022 and 2023 monitoring, and on the right side is our after fire, especially 2024, just 4 months. That monitoring occurred just 4 months after the fire, and then 2025 about a year and 4 months after the fire. And hopefully you can see that orange box is much higher in 2024 than all the others. And Phragmites actually really benefited it. I think it increased 154% relative to what the stem densities was before. So there was a ton more Phragmites stems. And I say Phragmites grows really densely, but this really, made me question how I say that, because it can obviously grow a lot denser when it doesn't have that litter that it produces. It doesn't have that litter to compete with. But now by 2025, it's kind of evened out again. The stem densities have dropped again, so there's much more similar stem densities than there was before the fire. The next question is, how does the biocontrol moths, how did those respond to the fire? And of course, during the fire, they would have been those really tiny eggs that were stored on the Phragmites stem, so just 1 meter wide and probably very prone to burning. And so initially in 2024, if we see that last bar, it's actually it looks like 0 because there's pretty much no biocontrol agent activity within the plot. So no damaged stems in our burned area for the most part. But by 2025, luckily, they've actually returned. Those moths were able to kind of return to the burned area and return to the patch as a whole, and damage even more invasive Phragmites stems than they were the year prior. And this photo is pretty telling. It's like a phoenix rising from the ashes. You can see in the corner of the photo here, one of the biocontrol moths, the caterpillar life phase of it, right next to one of those burned stems from the fire. So they did return. And so I like to say, not all is lost in the fire. Well, initially it was a very devastating fire for me personally, and also I really thought my experiment was over, maybe my PhD. But I was able to kind of shift my focus, and so Phragmites initially benefited from the fire. It was able to boost just a lot more stems. The effects were short-lived. It's kind of evened out again, but the impact of the biocontrol agents was also temporarily reduced, but now they've returned even hungrier than ever. And so that's what happened to the Phragmites. I will say a little note about what happened to me. I was able to, again, kind of reframe, and I did present this poster at the Canadian Botanical Association conference. I think it's a pretty funny title. It's What I Learned When My Field Site Burned Down, Exploiting My Tragedy for Academic Success. So I was able to, make a poster presentation about this, and it was a fun one to present. And I also presented this at the World Wetlands Day symposium at the University of Waterloo recently, and I won an award for it. So really, I think maybe a net gain has come out of this fire. And I think that's really everything I had to discuss today. A quick summary, although I know there'll be lots of questions, but invasive Phragmites is really a threat to wetland biodiversity and integrity, and luckily there are several options to control invasive Phragmites at all scales of Phragmites invasion. So in Canada specifically, we have an additional tool, these 2 species of stem-boring moths that were approved for use in Canada by the Canadian federal government to help suppress invasive Phragmites. So feeding by these caterpillars is very effective at reducing the health and the vigor of this invasive plant. And there's still that pathway, although we're very early on in the Phragmites biocontrol program, but we hope that this will disrupt the competitive dynamics between Phragmites and native wetland plants and ultimately allow for plant community recovery to occur. So start to see native wetland plants start to return to these invasive wetlands where maybe Phragmites kicked them out before. What can you do? I know this is a really active community group. Sounds like you guys are really involved with conservation, which I like to hear. But the biggest thing would be to learn to identify invasive Phragmites and maybe even distinguish it from the native Phragmites and help report those sightings. I was sitting in at the Invasive Species Forum yesterday and today and the day before. And I know that some groups actually use those iNaturalist data to help target where they're going to start controlling invasive Phragmites in their region. So those can be really helpful just to report, get the word out there that there's invasive Phragmites. Of course, if you're going out birding, if you're going out hiking, make sure you're cleaning your boots, your clothing, any equipment that you're using, or if you are working with machinery, make sure you're not transporting soil elsewhere. Really want to stop the spread of Phragmites. Any little bit helps. And I know you guys are already doing this, but keep cleaning up and protecting your local wetlands, especially the menacing wetlands. I know those are really important. So with that, thank you for listening. It's very rare for a PhD student like me to get a group of people who, are not really excited about Phragmites control, and I get to talk your ear off for almost an hour. So it's really a treat, and so I'm looking forward to any kind of questions.
Claire, we'll open up for questions. Now, why don't you stop sharing your screen?
Yeah, of course.
Lots of hand clapping going on. I'm just going to
Thank you,
pin
everyone.
myself here. So, very resourceful use of bad luck there.
Yes, exactly. Thank you.
Claire, one thing we are getting, I think maybe from your headphones or maybe with the jack, there's a little bit of electrical interference
Oh
happening.
no, I'm sorry.
But it's not, but I think when you move or your head moves around, sometimes there's a little crackle. So
Okay, I'll try to be really
keep
still.
perfectly still. And
I will.
like a stem of Phragmites, don't blow in the wind or anything.
Exactly.
Some questions, and again, I encourage people to either put questions in the chat, or you can raise your hand, and you have to raise your hand virtually so I can see it, and then we'd recognize you and you could unmute. But before if people do leave or anything, you might want to go into the chat and leave comments for Claire, thank yous, or wonderful job, all that type of thing, and we'll share that with Claire later. John Foster says, you mentioned 2 control agents and you're researching 1. What is the status of the second or the third? I'm not sure. There's 2 control agents currently being used?
Yeah, so there are 2 approved for use in Canada. Those are the only 2 that we've researched. but we have both of them approved, and I've been using both of them. So some of my field sites have one species, some field sites have the other species, and the University of Waterloo field site that tragically burned to the ground. That one, of course, was one that we were testing that has both species. So they're also our sites. But they coexist in Europe. We know that they're found at both sites, so it's not thought that they're going to interfere with one another. There's plenty of Phragmites to go around, lots of Phragmites to eat. So they are both out there in Ontario.
And John wants to invite you for a talk. I'll make sure John gets your email later
Yeah, please.
with the North American Native Plant Society. I'm the president, he says. And the way we get in touch, John, I'll make sure you get Claire's email later. We'll connect you two.
Yeah,
I'm sure
please.
Claire would be happy to oblige, right?
Yep.
And Eileen Power says, will the moths be able to survive winters in northern Ontario? And she has a secondary question, do Phragmites burn hotter than other dead grasses? Now, I know there was a little bit of talk about whether Tiny Marsh would be release site, and I'm not sure whether it was rejected because it might be too far north or...
Yeah, we actually have a release site in North Bay as well as other locations in northern Ontario. North Bay
And
is
are
the
the
one.
moths doing well
Yep,
that
they're
far?
surviving there. They've been there. I think they were released in 2023, and then as of last summer, they were still there and still eating Phragmites there. So they are from our source populations are mostly from Switzerland, as far as I understand, and Switzerland is also they're quite familiar with the winters as well. So I know it's hard to think about a little moth surviving, but they are well adapted to the cold weather and the harsh winters.
I'm just curious too. I mean, what the Latin name is something australis?
That's for Phragmites. The two moth species are Lanisa gemnipuncta.
No, but for
And
Phragmites, it's something
oh,
australis, right?
yeah, Phragmites australis.
And australis it's nothing to do with Australia.
No, it
Australis
means
just means from the south. But I mean,
I think it
most
means
people say
of the
this
west.
Phragmites come from Europe. It's just from southern Europe, or
I thought maybe australis in Latin translates to west, but now I'm second guessing that, so I'm not too sure. But I do know that they come from Europe, actually all over Europe, and actually Phragmites is one of the most well-distributed plants across the entire planet. It's on every single continent except for Antarctica now. So.
Bruce Boland says, have these moths been approved for use in the USA? Is it the same two moths that are being used in the USA, or?
Yeah, so they're still in the federal approval process. The first round of the federal approval process in the United States, the technical advisory group, they have recommended the moths for release. But I know there are some in the U.S. There are a little bit more bureaucratic hoops for them to jump through, and it's a really complicated situation. But they have not been yet approved in the United States, not because of any safety concerns or anything.
Now we have a couple of people that you've done a great sales job, because Pat Mitchener wants to know where she can buy some of these moths, and Sandy Agnew asks, are these moths available to the public for use on private property?
Not yet. I know we are still ramping up essentially production, but of course these moth populations take time to build. But my research collaborators from the University of Toronto, they've been doing a lot of really amazing work to get these moths on the landscape at 60 different sites in Ontario, and start to really amp up the production of nurse sites, which could eventually feed other sites as well. So not quite available for public use yet, but they are, getting out. We're trying to get them out there. We're also trying to raise public awareness about biocontrol, and so all sorts of things. It's a work in progress.
Again, John Foster asks, have the moths stayed on the invasive frag and not transferred over to the American frag? And again,
Yes.
if they're not, I mean, some of us have a hard time distinguishing between the two. How do the moths figure it out?
Yeah, so there's actually a number of safeguards in place for the native Phragmites specifically. So there are a number of differences. That's how we know that there's two. There's a native and an invasive frag. And so one of the biggest things is that the native Phragmites will drop its leaf sheaths in the late fall. And those leaf sheaths are where the eggs are stored. So when those if it were to, it's very unlikely that they would lay eggs from the testing that was explicitly tested as part of the federal approval process. But if they're, kind of in a freak accident, if they were to be laid on the native Phragmites, those leaf sheaths will fall and that will leave the eggs really prone to desiccation and predation. And so that's one way native Phragmites is protected. And then another way is that native Phragmites has a really small stem diameter. They're really skinnier stems. And so the moths are they're not too big, but they do require a certain size of invasive Phragmites to eat and to occupy and live on. And the native Phragmites seems to be too small for that.
Dorothy McCown asked, what has happened to the purple loosestrife moths? Have they gone to native plants? We don't hear as much about purple loosestrife, but how did that all work out?
Yeah, so the purple loosestrife beetles are still active in Ontario. One of my field sites, actually, I found them. And they still feed on the purple loosestrife. I'm not aware of any non-target effects. And what I do know is that over time, actually 2 decades have, or almost 3 decades by now, but 2 decades of research have come out suggesting that they actually do lead to native plants returning to these purple loosestrife invaded wetlands. And so really, the purple loosestrife is a really big success story for the field of biological control and have really astounding, long-lasting ecological benefits.
And Bob Codd, do you know how far the moths have dispersed now? They're not going to fly miles and miles, but...
Yeah, so the best estimate that I have is that, in 2 years at my field site, they traveled 2.1 kilometers. There certainly is more Phragmites past that I haven't searched, but we're continuing to research more on how far they're able to disperse.
And John Foster again asks, are there any endeavors ongoing to identify other frag pathogens from the other 58 you said might be available? I don't think we're looking to import more and more moths. We'd like to find a couple that will do the job, right?
Yeah, exactly. So I don't know of any active research. We're still very early on into this one program, and a lot of money's been invested. So now it's just a waiting game to let the moths do their thing, understand how they're impacting invasive Phragmites, and hopefully they're the only ones we need. But we never know. So
I know you mentioned Lynn Short in your presentation, and a lot of us know Lynn and have worked with her, and she. I think you could fill me in more of this, Kate, but she worked with Tiny Township and was getting a lot of students to get out there and tackle frag to get their volunteer hours. Is that right? You'd have to unmute. You have to press your little microphone button, Kate.
So Lynn does the cut
yeah, the
works
spading
in
technique.
sandy soil, basically. So it's really good along shoreline. And so, yeah, Lynn has been doing a lot of that all along the shores of Tiny Township with different groups.
Yeah, I've never met Lynn personally. I just, recognize her name as being cited, pioneering the spading technique. So it's good to hear that it's being used in Tiny, working to control Phragmites.
And of course, Kate was quite involved in Tiny Marsh and about a 5-year project on Phragmites there, right, Kate? Right.
Yes, I have a few comments and a few questions for you,
Sure.
Claire.
Yeah.
So just to explain, Tiny Marsh is a huge wetland. There's 800 acres of open water. And of that, there was about 325, as I remember, of frag that we had to tackle. And so we used cut to drown on the stuff that's in open water. And that was actually really great because it created all this biomass that we just piled up and made these wonderful islands that all sorts of birds nested on. And we almost regret they've now decomposed, but it was a useful feature for the marsh when it was there. And then we used Habitat Aqua for the stuff that we couldn't cut to drown, and we found that effective. It didn't completely eliminate the frag. We couldn't get at everything, but it certainly reduced it to, I think, about 95%. And we had good vegetation recovery, native dominated resurgence within the first year.
Wow.
So I'm really interested in your experience with the moth. I do have, just because of the law of unintended consequences and our history as a species trying to manipulate the environment hasn't always worked out for the best. So I am concerned about introducing a moth. I realize the horse has left the barn on that one, but I still have a concern. Um, and then my other thing is, are they really effective at controlling the frag? Okay, they can knock it back, they can make it wilt, it's not growing so well, the canopy's not so dense. But my experience with frag is that the root is pretty well just so strong. And you deal with the above ground stuff, but if the root system is still there and growing strong, you're not dealing with the problem. So I'm just thinking, you're saying the moths persist. The frag is a host plant for the moths, so they're happy together. But really, is that addressing the problem that we have in our wetlands?
Yeah, and that's a good question, and that's really what the core of my PhD is about. But the challenging thing to, I guess, convey is how long biocontrol takes. It's really about small changes over time. It's not anything like Habitat Aqua, where you spray it and hopefully 95% of it or more is gone the following year. It's about small, sustained changes to how the plant acquires resources. And so all those damaged stems, that's something that the rhizome, those resources, it's been invested to produce a single Phragmites stem. And so it invests a lot of energy, and when that stem dies, it's lost that, there's no gain on that. It's not photosynthesizing, it's not producing resources that can eventually be reacquired back by the rhizome system. And so the short answer is that's the hope, but we're still very early on in these phases. My research has only been going on for 4 years, and so we have early signs of success, but that we don't know if that's going to translate to really ecosystem-wide changes. But I like to remain a little bit optimistic, give the moth a chance, but also keep encouraging people to fight the good fight and continue to control Phragmites via the most effective method based on our evidence so far.
That answer your questions, Kate? I know.
Yes, it does.
Okay. Yep. Now, we're kind of winding down to maybe the end of the question period. If you got burning questions, get them in the chat. And before we leave tonight, though, I think we wanted to do our regular, like we do our nature sightings, and we'll open up the floor to anybody who has things to share. I think Bob Codd might share some of his wonderful photos with us, and he wanted to show some more photos about that over the Horizon Radar site. But I thought we had somebody on the talk, and I'm not sure if she's still here, Aya or Aja from Sudbury. And she sent us a little note before the talk. It says, I am from Japan. More old historical secrets are shared after COVID. Phragmites is one of the very hot topics in terms of making a strong boat. 30,000 BC, we know people were traveling from India, Vietnam, Taiwan, Japan in these areas by using the Phragmites boats. Ancient people were not in the dark. They were possibly smarter than us. We came to think this way because there were so many Phragmites remnants found in these regions, and the analysis suggests common wide trading older than 6,000 years. It's a surprise to know that they grow in these cold climates. And Aja said she'd love to know what kind of species are suffering with this invasion. Well, I think we answered some of those questions. But shows that, when it's part of the kept in its proper place, it was a useful plant to humans for many, many years. And if we can get it sort of in balance, I think Bob Codd said we imported the invasive plant, but we didn't import its competitors. And,
Yeah, exactly.
have to address that.
And I once did a talk, and someone from Europe said, "Oh, that's Phragmites in Europe. We use it for our roofs of our houses," and it was just it was such a different perspective than what we have here in North America. But the reason why they're able to coexist, with native plants in Europe is because they have all these herbivores. It didn't escape the herbivores like it did in North America. So very different perspectives across the world on the plant of Phragmites.
Yeah, that was something that I thought about. I was wondering whether we could use the frag from Tiny Marsh to kind of encourage people to use it for roofing or whatever. But we're not well situated for transportation corridors and so on to get the frag out to.
But it's nice we have been using it for black tern nesting sites. That's been a bonus.
Yeah.
Mhm.
Was
Yeah.
was wondering how well Phragmites would support a snow loader, personally.
A snowblower?
A snow load.
A snow
Oh,
load.
snow load.
Yeah, you get 1.5 meter snow on your grass roof. Is it going to hold up? I'd
Yeah,
rather
okay, I'd say you're talking about using it as roofing material. Okay.
I think it's pretty tough, actually. So.
Well, with that, if there's no more burning questions, I'm going to thank Claire. Do you want to thank her too, Bob? Let me pin you.
I'm going to thank Claire. I really appreciate this. You've helped spread the word. Personally, I'm a believer. This Phragmites is so widespread and in so many places, on private property, on public lands, inaccessible places. I just don't see any other way we're going to deal with it. And I'm reassured that these two moths aren't going to spread and attack other.
I might note too, for Claire's information, a couple of weeks ago, our Midland Garden Club had a presentation by Doug Tallamy. Now, do you know that name, Claire?
No, I'm not familiar.
Well, he's a famous author,
Entomologist. He's an entomologist.
entomologist and ecologist, and his books are very, very popular about native plants and kind of bringing native plants back. But he's a best-selling author. And I mentioned on the Zoom to Doug that we were having you come to speak about this biocontrol. He's from the States. He's a big American author. And he hadn't heard about these two moths, but he was quite excited about them. And I was fully in support of biocontrols and was dying to learn more about it. And hopefully that is successful. So thanks again for joining us tonight.
Thank you for having me, and thank you for the great questions. I'm always happy to speak to people, so feel free. If anyone has any follow-up questions, they can email me as well.
Is that Phragmites in your screensaver behind you there? Or?
Yeah, this is actually native Phragmites from Long Point. This is kind of the most pristine area I've ever been to is Long Point in the National Wildlife Area. They have both native invasive Phragmites, but they had already controlled the invasive Phragmites, and so there's this beautiful stand of native Phragmites, which I love to see.
Well, I don't know that we've got a ton of native Phragmites at Tiny Marsh, Bob, do we? But I know when we walk up our dike on Tiny Marsh, we can have invasive on one side of the dike and native on the other, right?
Yeah, that's correct. Yeah.
I want to give a shout out to Kate because she was the driving force behind this 4-year frag-free Tiny Marsh program. And it was an incredible success. It's virtually eliminated, although clearly not totally gone. And you're my inspiration, Kate. You know that. So big thumbs up to you. So
we had somebody just before you go asking if you could, were you willing to put your email into the chat so people could see that too for further questions or after the fact questions.
Yeah, I'll do that right now.
And with that, we'll say good night. I'm going to turn things, thank you again. We're going to turn things over to Bob, but he was going to kick off the sightings portion of the program.
Oh, was I?
You are, yeah.
Okay. Well, wish me luck. Somewhere here. Remember how to do it.
Now, if anybody has any sightings, again, you can either put them in the chat or you can raise your hand and unmute yourself and tell us all about what you've seen in the last month or two.
There, can you see that?
Well, you're still in presentation mode. You're not full screen.
Oh.
Might have to back up and start again.
I knew I'd run into trouble. Presentation mode.
Well, we're seeing your go full screen on one photo and see what happens.
You see that?
We see them all. Stop sharing your screen, Bob.
Okay.
And then on your own computer, get uh, one photo up ready to go. While you're working on that, maybe I'll do the little uh, one that I have.
I think that's a great idea. I can't do. Let me try. Don't go away on me. Let me try this one more time, and then if I
Okay,
can't
I'll
make it
stop
work,
sharing.
I'll give up.
We had this all perfected before the meeting.
And that's always how it goes. Share. So you're seeing multiple windows,
Yeah.
and now you're seeing multiple windows.
Double click to enter full screen mode. No, no, we still got all your photos.
You at least see that.
Not the way we want to see it.
Oh hell.
Stop
Okay, you go
sharing.
ahead.
Why not?
Stop
I'll leave
sharing.
it
I
to you.
think what you have to do is you have to get one photo up on your screen the way you want it, and then make sure that bring it up on your screen. But in the meantime, Eileen Powers had 2 raccoons this week: pileated woodpecker, red-bellied woodpecker, and John Lowry spotted 32 trumpeter swans on Moira Lake. Where's Moira Lake, John? And Claire has said goodbye. I'll share my screen and see if I can show you a few things. We're not the most technically proficient group, but we mean well. Okay, some of the things that I've seen in the last few weeks. This is the famous strong-barreled screech owl, and I caught him sort of half awake one day, enjoying the winter sun. He's got one eye open there. And some of my photos don't show too well on Zoom, but we've had a few evening grosbeaks around this winter. I think this was the one. I went out looking for the black-backed woodpecker that everybody saw. I didn't see it, but I saw about 50 evening grosbeaks in the same spot. This is actually Bob Codd's. We don't call them common red poll anymore. There's only one type of red poll, so they're all just red polls. And usually we see a lot of winters we get big flocks of these, but there don't seem to be too many around just yet this winter. Bob has just one who seems to have. This red poll seems to think he's a goldfinch. He hangs around with the goldfinches, although he fights with the goldfinches too a lot. But he's hanging around with the goldfinches at Bob's feeders. And this is over at Birch Street in Collingwood. I know they banned mallards there and tracked them, but I'd never seen a black duck with the transmitter on his back before. An American black duck that it's banded there on its leg and it has a transmitter on its back. I go to Kitchener Park in Orillia quite a bit looking for gulls. I went over there one day and I was expecting to knock off 3 or 4 gull species. Wasn't a single gull on the ice anywhere. And that's because there were about 6 or 7 of these guys around. And so all the gulls, I'm not sure where they went, but they went somewhere else. And down in Minising, I saw something. This is a juvenile bald eagle, and it's being harassed by both ravens and crows. And ravens and crows will attack each other, but if they've got a common enemy, they'll band together, like common enemy of the bald eagle. It reminded me of Mark Carney's comments about smaller nations banding together to compete with the big old bad American eagle. This is a kingfisher that spends the winter over where there's a little bit of open water in Kitchener Park in Orillia. And over in Collingwood, there might be more of these gather once the ice opens up there, but they're often Collingwood Harbour is a good place to see some of these canvasback ducks, which I think are one of the most attractive ducks. A friend of mine near where I live in Midland has this Carolina wren coming to her feeders. It's been there unfortunately, we didn't get to count this on our Christmas bird count, although it was around in December, but it didn't appear on count day. But I got this one in middle of January. Nice. Funny, I was reading Carolina wrens are not considered to be a migratory species. Where they are, they stay there all year round. But they're not that common in our area in winter or summer. And it seems if we do see them, it seems to be usually in the winter. So some of them must kind of move up here, whether we've got a lot more or more easily accessible feeders up here than down on the southern Great Lakes. And there's been a couple of great blue herons hanging around Kitchener Park all winter. Here's one, there's the two of them there. And Collingwood's a great place to see these long-tailed ducks, as long as there's some open water, which there hasn't been a lot recently. And that's, you can see why it's called long-tailed duck. There's two of the long-tailed ducks there. This is not quite their breeding plumage. The males are spectacular in their breeding plumage. And there's a little area called Birch Street in Collingwood, right by their sewage treatment plant that always has some open water. And there's often some unusual ducks there that hang around all winter. This is a female northern pintail. Most of the pintails leave our area when there's no. And haven't had a lot of these guys around this winter. Maybe more of them will come in the next few weeks. But this is a pine siskin that was coming to some of these feeders in Victoria Harbour. We get a fair number of these around in the winter, the red-breasted nuthatch. And I was able to count this bird on our Christmas bird count, but I went back in January and saw him again, a red-headed woodpecker. There's been 4 or 5 of these hanging around in our area all winter. I always kind of put it down to every 4 or 5 years, some of them seem to stay the winter when there's been a big mast year and lots of acorns about. Because red-headed woodpeckers, like the acorn woodpecker and like gray jays, they stash and store food. So they'll store acorns and crevices and trees and cache food for the winter. But then they also like to come to suet and bird feeders if that's available. That's the red-headed woodpecker again. There's a ringneck duck, again a duck that usually leaves us in the winter. This one's been hanging around the stormwater pond at the mouth of the Wye River. There's a suspicion that maybe it might be injured, unable to fly. When we go to Minising, we often look for these rough-legged hawks, called rough-legged because they have feathers right down their leg. And Kitchener Park, this is the waste site at Rural Waste Transfer Station, where the gulls like to hang around. And good place to see this. That pure white gull is a glaucous gull, one of the largest gulls we have, almost as large as a blackback gull. There's the glaucous gull in flight, surrounded by ring-billed gulls and herring gulls. And then not a great photo. This was at quite a distance, but the third gull from the right there is a lesser blackback gull, very dark black back, and not much bigger than the ring-billed gulls and the herring gulls around it. We haven't had a lot of these guys around, but there's a flock of them near Bass Lake and Orillia, the Pine Grosbeaks. I had about 50 of them over by Bass Lake there. I couldn't spot any of the males of the species would be a nice bright red. The females are more yellow. Although I think this one might be a young male because it's starting to show some red on the head and the tail. This red-tailed hawk I photographed at Kitchener Park, and I know some people may remember last February, February 2025, I had an adventure where I was able to rescue a red-tailed hawk that got trapped in the bushes, and we got it down to Shades of Hope, and we were able to re-release it in a couple of weeks. And whenever I see a red-tailed hawk at Kitchener Park now, I wonder if it's that same one from the adventure I had. So that's about what I have. Have you figured things out, Bob?
Probably not, but I'll give it another try.
Okay.
All right, share. 3.
Yep. Oh, that looked good. That's good.
Okay, awesome. So this is my red poll. And
your single red poll.
Well, we've actually had 2. A couple of times we've seen 2 together. But lately the weather's been warm and the goldfinches haven't been showing up and the red poll hasn't shown up either. This is the blackback woodpecker that you were talking about. It's kind of a rarity for this area. This is the bird that we traveled miles to Algonquin Park just to get a glimpse of. This one was right beside the road, a little forest concession road 8 in Oro-Medonte. So it was quite an exciting day to see this. There's another view of them.
Donna Lister says she's had an immature red-headed woodpecker at her feeder all winter.
We went to your place, Donna, to look, and your bird didn't show up. And as a matter of fact, you didn't show up either. So I guess we did knock, but you weren't there.
John Foster says he's got peregrine falcon, brown creeper, red-winged blackbird, mating long-tailed ducks at Thixon Woods in Whitby.
This in the middle of winter, birds willing to be photographed are few and far between. This is just a common tree sparrow, a North American tree sparrow, but I thought it was a rather good photo, so I thought I'd include it. This was exciting for us. People were with us during the chat. This is down near the old Site 41. Many of you know where that is. We've driven past these stand of old, mature, gnarly-looking trees dozens and dozens of times, saying to ourselves, what a great place that would be for a screech owl. And finally, this one. And
was that Piper I heard there?
No, it wasn't. Somebody else is barking.
Sorry, that was my dog. I'm muted.
Piper doesn't seem to care.
Okay.
And a snowy owl. Now, these are usually pretty common, and this was around the Tiny Marsh area. This is on Town Line Road. And usually you can count on seeing it 1, 2, or even 3 of them there. And if you go further down in the Menacing Flats, during eruption years, we've seen as many as 19 in a single day. But they've been scarce this year and last year.
You know, he's got an expression on his face like that fellow in Trump's cabinet, Kash Patel, who they always show with his eyes bugging out.
I'm actually proud of this bird. So this is quite zoomed in. He was on top of a silo, quite well back from the road. I'm going to guess about 100 meters away, and he wasn't facing me. And I quite softly, I made my squirrel imitation. And he turned around and looked right at me. Like, that's great hearing from 100 meters away. So I was really thrilled that he did this for me. So anyway, that's our snowy owl sighting for 2026. Excellent. This is the same site Ken was talking about, the Aurelia Kitchener Park. This is a North American mink. And this is probably the very same bird that Ken photographed at Bass Lake. I couldn't swear to it, but this is a pine grosbeak.
And you can see, go back to that one, Bob. They like to feed on sumac. So although he's sitting in a pine, his beak is all smeared with sumac seeds there. And there's a big, near Bass Lake, there's a big patch of sumac that was attracting them.
Right across the road is a huge patch, but when I guess it was during COVID, we had an eruption year for these things, and they were literally everywhere. And if you found a good stand of sumac, you were quite likely going to find these. That's not the case this year. If you want to see a pine grosbeak, you got to work a bit for it. This was again at Kitchener Park. This is a shrike. I would say it's a young one.
Well, Bob, wait a second, Bob. Breaking news. Go back to your screech owl photo.
Screech owl photo.
Is that way back there?
Yes.
John Foster wants to let us know that not only do you have a screech owl in that photo, you have Xanthoria periotina lichen.
Oh, well, I was really curious about that. I just knew it was kind of a yellow looking lichen. So we just on the one tree. Can you say that again, Ken?
Well, I think it's Xanthoria periotina.
Well, there's another nature sighting, and I didn't even know we made it. There you go.
Thank you
go. So now I've lost my place. Where was I? There.
On the shrike.
Yeah. Shrike. I'm quite interested by that. He's obviously a young one. An adult bird would have white on the top of the head. This is kind of gray. It's got a nice modeling pattern, and he was quite happy to pose for us. We weren't expecting to see him all, but he was the very first bird we saw when we arrived there. The same kingfisher that Ken spoke of, hanging around, chattering away in the stream. This is the discharge. Warm water from the Aurelia sewage treatment plant keeps the water open there year round, so species are able to survive there throughout the winter where they might not otherwise be possible. This is Ken's lesser blackback gull that he was speaking of. I managed to get a little closer to it.
Yeah, you got a Bob has a much longer lens than mine. And
you notice the difference here. He's got really orange legs, and that differentiates him from a lot of similar looking gulls. Gulls are one of those kind of birds that, we go looking for them, but just the thought of it makes my eyes roll back in my head.
now, I think you've got at the right side of that picture, you've got both an Iceland gull and a glaucus gull. Do you see that?
Yeah, I didn't include pictures of the Iceland gull, but yes, I'm pretty sure I did. And this would be the glaucus here. You're quite right.
And then the smaller white-headed gull beside that would be an Iceland gull.
This guy here?
Yeah.
I think that's a ringbill.
Oh, maybe you're right. Yeah, sorry. He looks so white.
Yeah, I think the Iceland gull had a kind of a smoky appearance on his face. It was there.
Yeah.
Oh, another photo of our mink. He was shaking off. It was droplets of water spraying everywhere. It was kind of neat. And this is at Washego. So it's a famous winter site for trumpeter swans. This is a red-breasted merganser. And
go back. Sorry, Bob. John Foster wants to chime in. Go back to your gull photo. He's counting 5 species of gulls in that photo.
Okay.
So what do we got? We got herrings. We got ringbill, lesser blackback, glaucus. I don't know that one. Would an Iceland gull have a ring? It looks so petite, that one.
This guy here?
Yeah.
No, I'm not 100%, but I'm fairly confident. I've examined all these photos, and out of the hundreds of gulls that were there was one very diminutive one. And not only I, but other birders identified it as a ringbill. I'm pretty confident that's a ringbill. At least there was.
No, some ringbills can be petite sometimes.
Yeah.
What's the 5th one, John?
I'm thinking it's on Iceland.
Which one?
The one underneath the glaucus.
This one here.
Yeah,
it would have a dark tip like that.
Yeah.
Yeah, it looks like an Iceland to me in terms of the shape of the head.
And the leg color.
These legs here.
Yeah.
Yes, you're absolutely right there. That is probably not a ringbill.
No. And there's nothing around the eye, and the bill is mostly yellow, and the color of the legs.
Good point. You got to come with me birding next time I go to.
Yeah. I've also got to become a member of MPFN.
Yeah, do that too. Do that first.
Yeah, which I will probably shortly after this video.
Oh, that'd be wonderful.
Yeah,
it's worth it. It's worth it. We got a new member tonight. Hooray!
Yeah.
And if you're under
that's our 5, but unfortunately.
Yeah, I had just one treasurer question. If a person renews now, when does the membership need to be renewed later this year?
Everybody renews on January 1st. So,
so it would be
join now. The next renewal date is January 2027.
Okay, great.
Yeah.
Okay. The mink again, the red-breasted merganser, and the iconic trumpeter swans. They were putting on a show, and I couldn't resist.
Well, everybody's been doing that at the Olympics, that heart shape,
Yes, I thought that was very apropos.
Yeah.
And here's one coming in for landing. Always like an action shot. They were very active, and I forget what the final count was. There was close to 90 trumpeter swans there, and 5 mute swans. And as the trumpeter swan people say, mute swans. Yeah.
And where was this?
This was Washego.
Washego.
We had actually.
Aurelia, there is an Ontario Mute Swan Society. I may get a presentation from them sometime. speaking up for these.
Well, I understand that the mute swans were a gift from Queen Elizabeth, if I'm not mistaken. And I know the trumpeter swan people wish she'd take them back, but I guess that's not going to happen.
There's also a trumpeter swan society here in Ontario.
Oh yeah, we hook up with them quite a bit.
Yeah.
But there's some people that speak up for the mute swans too.
Yeah, and there's also the trumpeter swan society in the United States.
And some of our members some of the members of the MPFN are also participants with the swan tagging program.
Down in the States, they use neck tags. Up here, we use patagial tags.
Anyway, this is going to bring us to this over-the-horizon radar array, and this is just a rather small sample. But can you imagine replacing productive farmland with 25 square kilometers of this type of radar array? And just to show you what it might look like from ground level. This is extensive. It's certainly alarming in such a valuable bit of farmland and such an important bit of wildlife habitat. So we're very concerned and looking forward to hearing more from the people who are actually building this. And this is the extent of the site. So the federal government has already required this block here in dark blue, and this is where their pilot project's going to go. But this extends from, what is this, concession 6 up to beyond concession 9. And the federal government is trying to buy up all this land so they can put radar receivers within these black outlined areas. And you can just, the enormity of the project is quite alarming. And I just wanted to share that with everybody so you'll know what.
Yeah, I think as I explained to us, Bob, when they did that blackout, it's not, because they don't really know exactly where they put it, but that's kind of the size that they would require. And there'd be 2 of those and it would go somewhere in that area. And it would basically completely change the nature of all that land around Strongville Road and right beside the menacing wetlands.
What I found most alarming, and I'm looking forward to confirming it with our upcoming meeting, is the suggestion that they need to grade these sites to within 2 inches of dead level right across the whole array. So that's an enormous disruption to the habitat, and that means denuding it of trees, existing buildings, everything else. It's unconscionable. I'm sorry.
And of course, once DND takes over an area like that, it's pretty well closed to everybody else. Nobody else can go near it.
If you're familiar with this area here, this is right adjacent. You can see it here. This is the menacing wetlands. There's a lovely shelter belt of spruce trees right across here, probably 4 or 5 trees deep. Their plan is to cut down all of those trees, replace them with a 4-meter high chain link fence, and keep the site lit all night long. I don't know. Perhaps it's absolutely essential for national defense, but I have trouble believing that there isn't a better alternative location than this.
Because we found out about this site down in Clearview. This is the receive site, right?
This is the receive site, and
the transmit site will be over in Carden.
It's going to look like this, and I think this is far smaller than their initial pilot area that they intend to build. So,
and in Carden, they're talking about taking over some of the road allowances. Well, they better not take over Wiley Road, although they'll have every birder in Ontario.
Well, personally, I think every birder in Simcoe County should be alarmed at what they're planning to do with this. This is one of our favorite haunts for winter birding. I can't think of any serious birder in Simcoe County who hasn't gone there to look for snowy owls and flocks of snow buntings and so on. And the fact that the drainage of this leads into the menacing wetland and what's to become of the runoff from a site that's been graded to 2 inches, within 2 inches of dead level.
The nice thing is that D&D sent letters to all the properties owned in the area. What do you want to sell to us? When you drive, almost every property has a no radar site. None of them want to sell. They want to be farmers. And so that's where this roar is coming from. The residents are up against it, and they're trying to get their local government to represent them, but we'll see how it all develops.
And that's all I have. And thank you for your perseverance and watching me struggle through presenting my few miserable sightings. So thank you.
So stop sharing there, Bob, and we'll say good night.
Say good night, Ken.
Anybody have anything else before we go?
It's Dorothy. Actually, I have a question for Bob because I went to see the trumpeter swans at LaSalle Park the other day, and I find the Washago swans seem to be friendlier because I had Griffin on the beach, well, and there must have been 100 swans just right by the parking lot, and they were like hissing at Griffin, like. So we didn't walk through. I don't know if someone had just fed them or something, but I find the Washago swans just ignore them completely. But of course, a lot of them are out in the river. But and I just wanted to say about the
well, the LaSalle Park swans, those are big city swans, Dorothy. They're not like
I guess so. They don't like all that commuting and crowding and everything.
Yeah.
Oh, and there was a dead goose there too that they weren't sure was avian flu or if it was lead poisoning.
Oh.
I was talking to some people there and they said, well, they hadn't had any, like the swans didn't seem to be affected, so they didn't think it was avian flu, but they had called Toronto Wildlife and, you know, to take it away, but they think it might be lead poisoning because the Canada geese tend to go deeper, I guess, and get more of the junk. And about the D&D site, because when we were there, I was checking it out too, and it's the fifth concession, which is where that church is, pretty much where the Calvary Baptist Church is, down to the seventh concession, which is where we usually turn when we do our Strongville Road birding hiking stuff. And so it's west of there, and then across the temporary site is south of that concession 7, and then the rest of it, the permanent sites are west of Strongville Road, crossing that County Road 10, which is pretty much like a highway nowadays, and
yes,
there will be 2 sets of fences, lights, and cameras, because they would still have to allow people to go down County Road 10. So yeah, there's going to be 2 sets of fences. And yes, I just happened to what the Roar people had given us the email addresses for the project manager, Irvin, and the NORAD people. And I just thought, oh, I'll just email them my questions. And they responded within a few hours. So yes, we have a call set up. Actually, did Sue check with you when you'll email me back, Bob, as to when you're available? Because we could probably soon.
I've been fussing around trying to prepare for this meeting, so I didn't.
Yeah, no, that's fine.
I didn't get
one of our questions will be, I understand that if they build this site, they'll have to close the airspace over it, and of course it's right beside the Edenvale Airport. So
this is true too. There's lots of things to be concerned about, and hopefully we get some answers. But Dorothy, while you're here, I want to thank you for following up on this, because I mean this is the original meeting with the Roar people was an eye opener. So this is a chance to get my questions answered. The particular concern is this idea that they want to grade this site to within 2 inches of level across the entirety of 25 square kilometers. It's just frightening.
Well, if you look at where the other sites are in Australia outback, and they're not near any farmland or cities or any green areas, it's like dead area. So yeah, that it's not a big concern in the outback probably, but here it would be. And you know, and it's really
Canada bought this technology from Australia.
But
evidently, that's not new technology, and a number of countries are using it. I found out that the United States has or is planning to have an array facing southward, monitoring South America. So the technology is quite widespread, apparently.
Question.
Yeah.
We'll
Question
hopefully
for
find out more about it next month.
question for Bob and you, Kenneth.
Yep.
Are you sending any of this nonsense to Ontario Nature and Nature Network?
Yeah,
And
I think we will eventually pull something together.
Once we get
Yes,
some
sir.
answers, I intend to, like, there's amongst birders, there's an app we use to share sightings called Discord. And I've had other things on the go, but I want to raise the awareness amongst birders, because I think a lot of people see the signs opposing the radar, but they don't really know what it is. And my experience, and I think Ken agreed with me at the time, was that quite often these military installations actually provide benefits for wildlife. For instance, the Camp Borden is habitat for the Kirtland's warbler, which otherwise wouldn't have. But once you see the reality of it, it's quite alarming.
Is there any reason why this can't be put on some part of Borden?
I think
Well,
people have suggested that, but see if we think the reasons are valid or not.
personally, if you know the menacing flats, it's the name is apt. It's quite flat, although not 2 inches within dead level flat. And I think that's just a very inviting site for them. It's going to make their job much easier to do the installation, rather than picking a site like Base Borden, which already has existing infrastructure that the government doesn't want to spoil, and also it has established pine forests. So
One reason we were given is that the Borden site has a lot of buried munitions, which would be expensive to clean up. Not impossible to clean up, but expensive. So
Yeah, and they tried to do the same thing with solar panels up to the east of Cardinalvar, and
Yep.
we all opposed it.
yes,
yeah. But that was a private for-profit company. This is the government of Canada,
Yeah.
which could be in the every
We still have to oppose them.
every time Mark Carney gets up, he's.
I agree with you, John, but the government's given itself the authority to ignore environmental regulations if the project's in the national interest, and I cannot foresee them not declaring this in the national interest.
Still doesn't mean we can't oppose them.
Yeah.
Oh, absolutely. I want more information before I jump in with both feet, but yes, as it stands now, I'm very concerned.
Now, Bob, do you think we should, like, I just invited you and Ken on this call. I didn't know if we wanted to add a lot more people. I didn't know if you wanted to, invite any more members of the MPFN, or if we just keep it small for now.
I think if you get a time set up, maybe we'll share it with the rest of the executive.
Executive, okay.
Um,
I had also sent the questions to Peter Burns. What's his last name? Anyway, a local engineer, Peter Burstein, who usually writes for Barrie Today, and I copied the Barrie Today off Kevin Lamb, who had put the article in Barrie Today. So I might ask if they can go on the call too. Because Barrie Today, they might want to do a follow-up, right? Because the picture that they had printed and that the Roar people gave us shows one row of those aerials, so it doesn't look like a big deal, right?
yes, exactly. They're deliberately misleading, I would say.
Yeah.
Keep up the good work, Dorothy. You and Deepthi are our best rabble-rousers.
No, sincerely, Dorothy, we really appreciate you following up on this and bringing it to everyone's attention in the first place. Very well done.
Well, it's my curiosity.
Okay, I think we better say good night. It's 9:30 past my bedtime.
Yep. We have to get up and watch the hockey game, 10:40. Well, be up in time for the hockey game. 13 hours.
Well, I have to get up at 8 o'clock tomorrow morning to watch the curling. So.
Oh, okay. Ooh. All right. Good night.
I hope you get a good curling fight.
Okay.
Anyway, thanks everyone.
Good night,
Thanks,
everyone.
Bye.
Stop the recording.
Bye.
Bye.
Bye, everyone.